Dr. Gruba.
Well, thank you very much, Justin, for the introduction and, of course, the invitation to talk to you all. It's really nice to connect with some of the other centers to learn more about what they're doing and to share some of our work so we can have some more connectiveness. I'm going to share my screen. I did it right. Okay, cool. Um, so, yes, I want to talk to you all about dengue virus, which is a major research focus in my group. Um, we I work with different groups around the world of trying to develop genomic surveillance programs for dengue to understand its epidemiology, to start studying vaccines. I think my good friend Andrea Morrison is going to present in one of the future ones from the Florida Department of Health, and I've been partnering with her for a very long time on dengue, and before that, Zika. I work with the CDC dengue branch in Puerto Rico, and have many collaborators in throughout the Caribbean and in South America and Asia. Um, and one of the things that my group really focuses on, and this is through the work with the PGCOE as well, is helping to create capacity, right? Taking some of the tools that we have and putting them out in other people's labs. And so that's primarily what I'm going to be talking to today about dengue, but also just to kind of set the stage of why this is important. So uh, as a dengue researcher, I'm contractually obligated to show you this figure, and that is that about 40% of the world's population is at risk to dengue, and that risk is changing, mostly expanding, and that's primarily due to the expansion of the vector Aedes aegypti. Where, you know, as you go back 20 years ago, and you did not see outbreaks in locations that you're seeing them now. So, for example, even in Florida, mostly in southern Florida, we're seeing outbreaks now in parts of Europe, even the southernmost parts of Brazil, northern Vietnam. We're seeing the expansion going north and going south as well of where we're finding dengue outbreaks. And it's not just that dengue is going to new places. Um, Also, in some of the places where we have dengue transmission, it's been increasing. So from 2014 to 2023, this is what dengue cases in the Americas look like. And you see where you go through patterns of high years and low years. And in the last 10 years or so, we saw a lot of record-breaking years happen. And 2023 looked really big. And it's like, okay, now we have the most number of cases we've ever had. And 2024 comes along and just completely shatters that. 13 million cases reported just from the Americas. Most of those were in Brazil, but we did see large outbreaks in many other places. So this is a growing concern, and we need to have better tools to be able to deal with this. A very major component of what makes dengue what it is that there's four distinct serotypes. These are not only genetically distinct, within serotypes, there's a lot of genetic diversity that can have phenotypic variability. But the primary part of this, of why we care, is that infection with one serotype doesn't necessarily mean that you have lasting protection against other serotypes. There's kind of this Goldilocks scenario where if you have really strong antibodies to one serotype, you might have some short-lived protection against other serotypes, but as those antibody titers start to wane a bit, you can actually create antibody-dependent enhancement. So secondary infections are often more severe. And this aspect of ADE, antibody-dependent enhancement, and secondary infections is really what allows for the continuous co-circulation of multiple serotypes. But because we have four distinct serotypes, all of our control measures need um, to account for this, in particular vaccine development. So there's three primary vaccines that have been in development. We have the Sanofi vaccine, Dengvaxia, We have the Takeda vaccine. I have it as its scientific name, TAC-003, but it's also called Qdengue or Dengue. And then we have the Merck Butantan vaccine. All of them attempt to be quadrivalent, meaning incorporating at least part of the external envelope protein of all four dengue serotypes, and they do so in slightly different ways. And this has led to some mixed results when it's come to clinical trials and the real world rollout of these vaccines. So Dengvaxi was the first one to come out. Um, this is really a yellow fever vaccine that we've been using for a long time as one of the most successful vaccines for yellow fever, and using that as a backbone to switch in dengue 1, 2, 3, and 4. It turns out that it's not enough of dengue to really make it a great vaccine. It requires 3 doses, which is really hard to administer. And it turns out if you are seronegative and get the vaccine, you don't really have much protection against disease. And in some instances, they have noticed enhancement of severe disease in young kids in a clinical trial that was in, I think, Indonesia. And so when it was licensed in 2015, it had this giant caveat that you had to be seropositive to get the vaccine. And so in Puerto Rico, where they have attempted to roll this out, I think only some like 200 or so doses ever got administered because it's quite complicated where you have to go in and have your serostatus checked and then get 3 doses of the vaccine. And it still wasn't all that great. The Takeda vaccine has been much more successful. This is now licensed and it's being rolled out in several countries. They're still trying to figure out what countries are going to start to buy vaccines to subsidize it for its population. It's kind of expensive to buy it on your own. Mostly being rolled out right now in Asia, I believe, though there was some usage in this past year in Brazil. Um, it's it has much better effectiveness. It's only 2 doses, but it's really great for dengue too, and not quite as effective for the other serotypes. Um, the third vaccine that is being co-developed by Merck and Butantan, this was actually originally developed by the NIH. This one is a single dose vaccine, and the clinical trial results for this have been really good so far. And unfortunately, the formulations between Merck and Butantan are different enough where they have to do different clinical trials. Um, but we're expecting in the next couple of years to come that this will probably also be licensed, and it might be the best one available. But so we have some imperfect vaccines, but we have some good options on the horizon, which are going to be really important. But because we have 4 different serotypes and we have a lot of genetic diversity within serotypes, we need programs that can evaluate the real world effectiveness as this comes out in the context of viruses that are circulating. So virus genomics is a great tool for this. We saw this with SARS-CoV-2. I probably don't need to belabor this point of why that was important to be able to know what variants were circulating, because then we can get variant-specific effectiveness for vaccines. We use that to update the vaccine formulations. I don't think it's going to be quite like that for dengue, where they need to be updated, but we need to have a better sense of what viruses they work well for and when they don't. So if vaccine effectiveness does start to decrease, we have an explanation for it and we can explain that to the public, and it doesn't create mistrust in the vaccine programs themselves. Um, and so, we would love to have systems in place where, people who come in that are hospitalized, we're taking samples, we're sequencing them, doing bioinformatics to inform a lot of different things when it comes to diagnosis and treatment, and use those data. The pie in the sky part is for individualized medicine. We're way, way far away from that, but we can certainly use the data for population health. Um, Another way that my group works with public health departments is also to use these genomic sequencing data um, and phylogenetic analyses to understand patterns of spread. What viruses are circulating? Where do they come from? How long are they staying there? What populations maybe are they spreading in? So these trees here are what we call phylogeographic reconstruction. And that is because all the tips of these trees, all the colors there are different locations, and we have one for each of the four serotypes. And the branches are colored by locations that we infer through the phylogenetic process of where the ancestors of those viruses existed in time. And so when we have time and location that is estimated throughout these phylogenetic trees, we can do some really cool things like we can map them, we can find out where they go. And so I'm going to show this fun little video here going back of this is dengue 2, looking at the initial emergence of the virus in Asia and how it spread to the Americas and spread to Africa. And then you see a lot of within region spread. And occasionally you have these big transcontinental jumps of the virus. All the colors are different lineages or variants of the virus that I'll talk about in a bit. So you can see that there's sometimes separation of different genetic diversity, and then sometimes there's mixing when you have jumps between regions. And so a lot of times what my group is interested in is these jumps. When you have a virus that hadn't been there before that comes in, right, and is now spreading, does our existing immunity to other similar viruses, how well does that work for these new viruses? How antigenically different are they? Um, so this is what we want to do. And now I want to take a step back and talk about, to do the work that I was just describing for dengue, we need a lot of tools in place and things that are easy to use for public health labs. Ideally, this work isn't always being done in research groups, but is done for routine surveillance. And so how do we get these things in the hands of the people who need them? Um, the main thing that we can do here is we can tap into the capacity that was built around COVID-19. So this is from a review that we published a couple of years ago, but I love these figures, not necessarily for showing, the millions of viruses that were sequenced around the world, but showcasing the number of labs that have started to do sequencing and where they existed. So in 2020, when sequencing for SARS-CoV-2 first got off the ground, it was still really impressive the amount of data that's being generated in all the different places. But you notice in gray here are countries that did not have any of their own local sequencing, and there were a lot of gaps, particularly in Africa. You fast forward a couple of years, and the balance is still not perfect, but we filled in a lot of those gaps, meaning that sequencing is now being done in so many places that had never been before. And the cumulative number of labs that had actually sequenced viruses or SARS-CoV-2 by the end of 2022, anyways, approached almost 4,000. And that's just really incredible. Um, and what is really helpful in this case is that most labs are using the same base protocol, maybe different slight variations of it, but it was basically the same thing. That assay was something that my group actually helped develop way back during Zika. And there was some earlier development of it actually during the Ebola epidemics. And it's really just using multiplexed amplicon sequencing. So you design little PCR primers all over parts of the virus and you split them up into two non-overlapping reactions. And for pretty cheap and relatively easy framework for the molecular biology lab that you can amplify the whole genome and sequence it. And what's really nice about this protocol is that you can adapt it very easily for other viruses. So what we have been trying to like basically shout really since sequencing around 2023 and into 2024 for SARS-CoV-2 really started to decrease was that let's not turn those systems off. Maybe we don't need them as much for SARS-CoV-2, but let's point those systems to other viruses or even bacteria that really could benefit from genomic surveillance. And dengue, I think, is a very important one to add to this list. So basically, let's keep our partnerships that we have with the clinical systems that are collecting samples, that are collecting metadata, right? We have IRBs that are already in place to do this work, or for public health labs, you already have the relationships of sending those samples to your labs. In the wet lab, we have the base steps in place. We have the PCR amplification, the library prep, the sequencing. My kids love Legos, so I think about that a lot for this. And For the primers then to switch to different viruses is just like switching out a Lego color, right? You change a few base things, you change the primers, and then in the bioinformatic end of it, you just have to change a few files and you can run your same SARS-CoV-2 platform for other viruses. And we've done this for many different things. We help the Florida Department of Health labs switch from SARS-CoV-2 sequencing to dengue sequencing using our protocols. We've developed taking the COVID-19, the COVID-seq protocol, and we've used it for Mpox. We actually are using it for bacteria now to sequence TB, and we're using it for RSV and HMPV. This is the major part of our activities within the Pathogen Genomics Center of Excellence, is just creating a whole bunch of different protocols that you can tap into using the COVID-seq pipeline. Um, so for dengue, it was a little bit more challenging to develop these protocols in the sense that you have four viruses. And what you don't want to do is take time before you do sequencing to figure out what serotype it is, and then do a serotype specific sequencing protocol. There's a couple of reasons why that's not a great plan. One is it adds a bunch more time, right? And it adds opportunities for things to be less efficient and go wrong. But also it could create biases in your sequencing themselves. If um, don't have many dengue 4 samples, right? Then it takes you longer to fill up those runs. Maybe you don't do dengue 4 sampling or sequencing at all, and you end up missing those and they don't go into our databases and they don't go into our systems. What we developed is once we know that it's dengue, doesn't matter the serotype, what we do is we mix all the primers for the four serotypes together in the same sequencing assay, and they work without interfering with each other. So we were able to create this pan-dengue virus sequencing protocol to reduce all of these biases and save time and all those good things. Um, the protocol itself is available on protocols.io. We have primers that we give people if they want to start sequencing on their own, or they're pretty easy to purchase for many of the places that you normally would get them. We also created a bioinformatic pipeline that goes into this. Again, it's pretty similar to if you're already doing SARS-CoV-2 sequencing, but we added in a few updates because for dengue, since there's four serotypes, what it does is first is it figures out from your sequencing data what serotype that it is, and then it goes in and aligns those reads to create those genomes for processing. And we also have a option if you like to do your sequencing on nanopore versus Illumina, there's methods there for that. So we've tried to make this end to end where you have everything that you would need to start sequencing and analyzing your data. So creating genomic data is one thing, right? One of the things that I learned from working with public health labs for a lot of different emergencies now is if I sequence viruses and I just put them all in a phylogenetic tree and I'm showing that to somebody and they don't have a whole lot of experience with phylogenetic trees, there's kind of like this like really negative reaction to it of like, what am I supposed to be getting out of here? And oftentimes, it's not the phylogenetics that is really important for what we're trying to do in an applied public health setting, but it's really the categorization of the genetic diversity that we have there. So, for example, in SARS-CoV-2, we had named variants, we had lineages that had all sorts of numbers and letters with them. But it allows us to take, because we have a collective understanding of what classification of genetic diversity within the phylogenetic tree means, we can use the nomenclature based on the specific mutations and patterns of mutations and lineages, and we can look at temporal distributions of those lineages to see what's or of those variants to see what's increasing over time, where they are at spatially, and then we can start to apply some more basic or classical epidemiological things like we can estimate the RT, the growth rate, right, of these things. We can apply this to clinical studies where we can then look at variant specific effectiveness and get rid of the genome itself and just use the classification. This works great when you have a system that we all say, okay, I agree with. For dengue, we did not have one of those. Classically, or at least since the early 90s, within serotypes, we had some roughly developed genotypes of dengue virus, usually about 4 or 5 um, per serotype. And these have some historical segregation in geography. So, for example, if you were in South America sequencing dengue 1, right, the genotype didn't really give you any more additional information because all dengue 1 in South America was this genotype 5. And so if you can serotype the virus, why would you sequence the virus? Because you're not going to get anything more. But really, there's a lot of genetic diversity that exists within that genotype that might be important for, again, understanding vaccine effectiveness or introductions and spread, persistence and locations. So we worked with um, groups from uh, 14 different countries that study dengue virus to come together on a system to reclassify dengue virus into lineages and major lineages and minor lineages. This is not so crazy like SARS-CoV-2, where we had aliases and several dots and it went on and on and on forever. These things are a little bit more stable. We have some pretty conservative rules for calling new lineages, but it allows us to take, so here showing dengue 3 and break it up a little bit further into major lineages, further than that into minor lineages, where we come to a sweet spot of it's precise enough where we can detect when something new comes in, but it's not so detailed and it's not so subject to change that you would just have hundreds of new lineages that are emerging every single year. So we developed the system, and what it does now is, so here's an example of dengue 2, genotype 2, which actually has a global distribution. If you look at major lineage A, it's mostly found in West Africa and uh, or sorry, East Africa and parts of Asia. And then we look at these two different sublineages of A2.1 and A2.2, and they have some overlap in their distribution, but also some unique places. So, for example, then, if you were sequencing in Brazil and you detect this lineage, the moment you find it, you know that you have a new introduction because we had not seen this lineage there before. You do not need to have a phylogenetic tree to get that insight. And that's where it really is helpful. Also here, so this is an example looking at dengue 2 as well from Vietnam. And there was a big outbreak of dengue 2 in 2012 and also in 2019. Um, Both of these are actually of the same genotype. And so if we didn't have further information, we would be like, oh, this is really interesting. 7 years apart, we have an outbreak that's caused by the exact same genotype that is here. But actually, when looking at it, we see it's a different sublineage that has emerged that caused the second outbreak, which allows us to then start thinking about some questions around, is there enough antigenic difference between these that the people that were infected here were no longer protected here? Um, So we launched this website called denguelineages.org last year, which has a whole lot of information on the design of it, our assignment tools, which I'll tell you about in a second, a bunch of resources and such. And we have a scientific committee consisting of people that really help to build this and experts from around the world. Verity Hill, who is the technical expert around this, she's now in Belgium. So the five of us represent 5 different countries. And we make it really easy for people to get a hold of us if they want to learn more about our system. But one of the things that it does that we did within this is we created assignment tools. So if you're doing your sequencing and you have your data, how do you determine what lineages you have? A lot of people who work in the dengue field use this tool called Genome Detective, so we have that available. But Nextclade is one that we really like. So if you go to our website, you'll see a little link for it, and you just click it, and it'll take you there. You upload your sequencing data, and it gives you a file like this. And it's nice, there's some quality control results in here that you can look at here under clade. This is your lineage that you get from this. And you can see the coverage of your genome and other sorts of things. And this is the results file. If you click over here on this tree file, you can actually see where your data fits within a subsampled base phylogenetic tree. So you can see that from these dengue 2, we actually have two different genotypes of 2 and 3, and we have three different lineages in total, and you can see where they exist here in the phylogenetic tree. And if you were had a question about one of these, it's like, okay, what is this lineage? I have one of these here. I've never seen this in my data before. You can go back to our website and there's a tab at the top that says lineages, and you can flip through this and you can find the major lineages. You can find where this is and click on it, and it shows you all the current data. And it shows you where we currently have sequences for it, how it spread to other locations. The data that I was just showing you was from Colombia, and this would have been, or this was the first ever detection of this lineage in Colombia. So once again, just by going through and looking at this and using our tools, we already know that we have a new introduction of the virus there. Some previous work showed that this um, lineage actually has some mutations that make it resistant to some developmental dengue drugs, and so it's certainly one to watch. We also have a resources page where it gives you links to data that people can use, our PCR assays, our sequencing assays, bioinformatic pipelines, assignment tools. Here, these are things for phylogenetics, where to find data. We're ever trying to grow this, but we're trying to make it a one-stop shop. If you are trying to set up dengue virus genomic surveillance, all of your tools are here. Okay, so all of that was just to say, here's the work that we did so that we can actually develop and start using these programs. Now I'm going to talk about a couple of applications, and I'm going to talk about the applications with some of my good friends in Florida and also with the CDC on a couple of different ongoing projects with some folks that were in my group and obviously with the Andrea's team and the CDC team. So the first, we're going to talk about travelers coming into Florida because this has been a really important source of information, not only for um, the dynamics of dengue in Florida by where the people are getting infected from, but also to allow us to look at those source locations of where dengue transmission is happening, and then to investigate local cases. This is really what our major part of the collaboration. So this is all from a paper that we published last year. And this looked at travel-related dengue cases reported in Florida from 2009 to 2022. And you see that we get travel cases from a lot of different places, but the majority of them come from the Caribbean, which makes sense based on their close locations and travel volumes and everything. And Cuba sticks out the most. And again, there's a lot of different connections between Florida and Cuba where this makes sense. But still, that number was a lot. And we weren't expecting it to be so much coming from one location, which prompted us to investigate this. So what we first did is we looked at the correlation between local and travel cases. So local cases in the source country versus the travel incidence rates that we estimated of people coming into Florida and getting diagnosed with dengue. And we found from our travel data and then the local data from Dominican Republic and then Jamaica and Puerto Rico that these mostly lined up, that our travel incidence rates really matched local transmission that was reported, which made us feel quite good about the data that we had and everything. That, of course, the more transmission you have in a location, the more likely that someone visiting there or traveling from there would have dengue. Where that doesn't align as much as in places where the local surveillance is not as good. So Haiti historically has not had great surveillance for infectious diseases due to a lot of different factors, and Cuba as well. I actually think that surveillance in Cuba is actually probably quite good. It's more of a reporting thing. But when we don't have data that we can use that is being reported, it's hard for us to get a sense of what is actually happening. So we created this model that was calibrated around the local and travel cases from the Dominican Republic, Jamaica, and Puerto Rico, and applied that to Cuba and Haiti so that we could try to estimate local transmission rates in those places per year. And then what we did is we took those annual incidence rates and we plotted those with other incidence rates that were reported from other places. And all the dots represent outliers, which are relative to the incidence rates of that countries would represent big outbreaks. So you can see here there's been quite high incidence rate outbreaks in these countries. 2022 Cuba is right here. This was what we're estimating as a very big outbreak. And you can see where Puerto Rico and Jamaica and other places fit into that. So this year, this in 2022, the reason why we had such a high number of cases of travelers coming in from Cuba with dengue is because they had um, relative to the last 10 years in that location, a really high, really big dengue outbreak that was unreported. Being a sequencing lab, of course, we sequenced these travel cases as well. Um, the vast majority of the sequences from Cuba was from this one clade, this single lineage here of dengue 3. And then diving into it, what we found is that this represented a new introduction from Asia into the Americas. Um, so we estimated that this emerged in Cuba probably around 2020 and rapidly spread in the island and then went to subsequently to many other locations elsewhere in the Caribbean. And I'll show you in a bit. It's gone even further than that. We also detected in Jamaica, first only through the sequence travelers, but now new sequencing from Jamaica itself also places more genomes here within this clade of another introduction that came in from Asia into the Caribbean, but didn't necessarily take off as much as this one did here. And in fact, we think that this is now probably extinct in the Americas. But this new lineage is now the main thing that is transmitting of dengue 3 throughout the Americas. So within a few years, this is spread all over the place. Um, dengue 3 is a really weird dengue virus that it goes through periods of about 10 years or so in between big outbreaks that we see in different locations. And we're definitely in a period now of the resurgence of dengue 3. Some of it is dengue 3 that existed in the Americas before, but a lot of it, um, especially in the Caribbean area and Central America, is this new lineage. And again, this is prompting a lot of different questions about the antigenic diversity of this virus and if people who have been exposed to dengue 3 before, are they not protected against this? Is there some additional enhancement from ADE that is going on, leading to more severe cases? A lot of different things that we don't know yet, but we needed the surveillance system to even detect that it was there. Um, so switching to looking at Florida then for a second, here is data from the Florida Department of Health that clinician in my group put together, just showing the history of dengue transmission in the state. I think Andrea will probably talk about this a lot more. This just showing that it's really um, the southernmost counties that have the most transmission, with Miami-Dade being near the top. You've had a lot of different outbreaks that have happened, but in recent years, the number of cases in Miami have started to increase quite a bit. And to give a lot of it away here is that this increase that we started seeing in 2022 and into 2023, and we don't have 2024 data on here, but that was also a big year, was really coupled to what was happening in Cuba. So here's an investigation that we helped Florida and the CDC team conduct. Um, and this was looking at travel cases versus local cases. The travels in blue, I've already told you about all of these. Most of these are all from Cuba. Most of the travelers are being diagnosed in Miami, and that's also where we saw the highest number of local cases. The virus there was dengue 3, and it was of that lineage that I was just mentioning. And so here, this phylogenetic tree shows just the sequences that are local to Florida. The orange in the background here, these are all Cuba. And so you can see that from Cuba, there's several um, uh, independent local transmission chains that have happened really throughout 2022. Some of these clades are a bit larger, suggesting that there maybe is some more secondary transmission that happened, but most of these are what we'd call singletons. So just one sequence that we have. And so it could be there was one case of local transmission, and then those transmission chains died off. Um, of course, we don't have all of the infections here, so we can't say that that is exactly what is happening, but it implies there's not a lot of secondary local transmission. You fast forward to 2023, and actually now instead of all of these small transmission chains, we actually see that the cases are concentrated more into these bigger clades, suggesting there is actually more local transmission happening within this year. Um, and then in 2024, weren't able to sequence as many cases, but we're also seeing these small clusters. And what we've been monitoring here, importantly, was these uh, transmission chains that you have in one year, and then you see reemergence of transmission the next year. Are they actually related? Meaning, do you get overwintering of the virus or trans-seasonal persistence of the virus between 2023 and 2024? And so far, we haven't seen that yet. And that is important because that distinguishes what would be like hypoendemic, or what I like to call summer dengue, where you have travelers coming in and sparking local transmission and then it eventually dies off, versus endemic transmission. And why this is important is because summer dengue might be a couple hundred cases. Endemic transmission could lead to tens of thousands of cases and a whole different sort of public health emergency. Dengue being endemic to Florida would be quite bad and a huge sort of stress on the public health systems. So we're doing a lot of research to understand if this can happen, and if and why it's not yet happening, and what would need to change for that. And we're also doing the sequencing and working with them to actually monitor this in real time to see if we start to find evidence for some transseasonal persistence and maybe the early makings of endemicity, which again, we're not seeing yet, but it's something that we need to monitor. And really, the primary way to monitor this is through sequencing and doing analyses like these. Um, the last thing I just want to talk about is looking more about what had happened here in 2024. So I don't just work in Florida. Like I mentioned, I work in a lot of other places. Here I'm just showing some data from a collaboration that I have in Cali, Colombia, where we're setting up a longitudinal genomic surveillance program, so that we have something in place for when dengue vaccines roll out, where we can actually monitor dengue vaccine effectiveness. This needs to happen in an endemic zone, and especially a place that has very high levels of transmission. And Cali, Colombia happens to be one of those places. 2023, 2024 was a very big year for dengue in this region, the most number of cases that they've ever reported. Typically, dengue outbreaks are driven by like one dominant serotype, and then sometimes you get, a little bit of the other serotypes. But you don't normally see something like this where you have like the co-dominance of multiple of them. And within these serotypes, we actually have several distinct lineages. And so, one of the things that we can say here is that the massive number of cases in 2024 was not due to a new virus coming in and a variant like what we saw with like Omicron and other sort of SARS-CoV-2 variants, where you have a new thing that comes in and this huge wave of cases. That's not the case of what driving dengue. In particular, we are concerned about this lineage because this is that emerging lineage that we saw in Cuba to Florida, and then started to spread. That this was not really even common here in Cali. It wasn't even the most common dengue 3. The most common dengue 3 that we found was this lineage C.1 that actually had been circulating within South America for the past 20, 30 years. So this was the reemergence of something local versus something new that was actually brought in. So we can kind of conclude from this is that the huge number of cases was not due to the virus, it was due to other factors. Uh, and some of our other data here that I'm not showing points to is because it was a big El Nino year, and there's such a strong relationship between temperature and dengue transmission that it was just perfect for transmission to occur. And all the lineages that were there had ample opportunity to increase transmission and to cause infections and to be captured in our surveillance system. So some parting notes that I will leave you with here of what is the future of incorporating dengue virus genomic surveillance into public health, but you can swap out dengue for many other viruses here in this situation. But the things that we want to do is we want to make routine sequencing of viruses and other pathogens a core function of what public health does because of the things that I think that it can do for public health, like have us make predictions. You know, when you have a new lineage that emerge, how long do we think it'll take to cause an outbreak, and where will it spread next? And we're working on methods right now to incorporate genomic data into seasonal forecasting to help improve our estimates of what transmission either in the near future or the longer term future, like next year, what that will look like by incorporating information about the circulating genetic diversity. A lot of different groups that I've worked with here to think. Um, there's a lot of different people of my group that have come and gone that have worked on these projects. A lot of these things started with SARS-CoV-2 and kind of expanded from there. And I'd like to thank my funding, my DP2 from the NIH. Our dengue lineage team is growing, and we're trying to find new funding to keep that program intact. My contact information is up here if you would like to send me anything, but I'm also not hard to find on the interwebs. And I will take any questions that you all may have. Thank you.